Number 1. How do monosaccharides produce acidic sugars? Monosaccharides can produce acidic sugars through oxidation reactions, where certain functional groups like aldehydes or primary alcohols are oxidized to carboxylic acids. This oxidation process can occur under certain conditions, such as in the presence of specific enzymes or chemical oxidizing agents. The resulting acidic sugars can then contribute to the sour taste commonly associated with certain fruits or fermented foods. Number 2. What are the 3 acidic sugars that monosaccharides can produce? Explain their difference. The 3 acidic sugars that monosaccharides can produce are the aldonic acid, aldaric acid, and the algironic acid. In aldonic acid, weak oxidizing agents oxidize the aldehyde end of an aldose structure to yield aldonic acids, while in aldaric acid, strong oxidizing agents oxidize both ends of a monosaccharide, yielding a decarboxylic acid called aldaric acid. And in algironic acid, enzymes oxidize the primary alcohol end of the monosaccharide without oxidizing the aldehyde end to yield algironic acid. Number 3. What is the process involved for the monosaccharides to produce sugar alcohol and what catalyst is involved? The process involved in the conversion of monosaccharides to sugar alcohols is called reduction. This reduction reaction typically involves the conversion of a carbonyl group, aldehyde or ketone, in the monosaccharide molecule to a hydroxyl group, resulting in the formation of a sugar alcohol. The catalyst involved in this reduction process is a hydrogen. Certain reducing agents like hydrogen gas are often used in conjunction with a catalyst to drive the reduction reaction to completion. Number 4. What is the general name for monosaccharide acetals and how do they form? The general name for monosaccharide acetals is glycosides. They form through a reaction between a monosaccharide and an alcohol in the presence of an acid catalyst, typically under acidic conditions. This reaction involves the substitution of the hydroxyl group OH at the anomeric carbon of the monosaccharide with the alcohol group from the alcohol, resulting in the formation of an acetal linkage. Glycosides are often used as storage forms of sugars in plants and have various biological functions. Number 5. What are oxyacids and how do the reaction with monosaccharide end up to? Oxyacids are acids that contain oxygen, hydrogen, and at least one other element. When oxyacids react with monosaccharides, they can lead to the formation of sugar acids or acidic sugars. This reaction typically involves the oxidation of certain functional groups in the monosaccharide molecule, such as aldehydes or primary alcohols, to carboxylic acids. The resulting acidic sugars contribute to the sour taste commonly associated with certain fruits or fermented foods. Additionally, oxyacids can participate in esterification reactions with hydroxyl groups in monosaccharides, leading to the formation of sugar esters. Number 6. Monosaccharides can form amino sugar. Explain how. Monosaccharides like glucose or fructose can change into amino sugars through a process where they get an extra part called an amino group NH2 added to them. This happens through a series of steps in the body's chemical reactions. For example, glucose can turn into glucosamine, which is an amino sugar, by adding that amino group. Number 7. Explain the significance of superimposable and non-superimposable mirror images in the concepts of chirality. In the concept of chirality, superimposable and non-superimposable mirror images play a crucial role in distinguishing between chiral and achiral molecules. The significance of understanding superimposable and non-superimposable mirror images lies in the recognition of chirality, which is fundamental in various fields such as chemistry, biology, and pharmacology. Chirality influences molecular interactions, reactions, and biological functions, making it crucial for drug design, asymmetric synthesis, and understanding molecular recognition in living organisms. Number 8. Distinguish stereoisomers, enantiomers, diastereomers, epimers, and anomers. Stereoisomers, it does not differ in their atom-to-atom connectivities, but only in the orientations of atoms in space. In enantiomers, these are stereoisomers whose molecules are non-superimposable mirror images of each other. In diastereomers, these are stereoisomers whose molecules are not mirror images of each other. In epimer, these are diastereomers whose molecules differ in their configuration at one chiral center. And in anomers, these are a special type of epimers that differ in their configuration around the acetyl or anomeric carbon in cyclic forms of sugars. Number 9. How to determine the handedness of a monosaccharide in both Fisker and Howard projections. To determine the handedness of a monosaccharide in Fisker and Howard projections, it involves examining the orientation of the hydroxyl groups around the chiral centers. In Fisker projection, assign priorities, lowest away, check arrangement, and in Howard projection, visualize cyclic structure, determine orientations, assign configurations using same priority rules as Fisker. Number 10. What are the classification of carbohydrates? How are they classified? Carbohydrates are classified based on their chemical structure, size, and functional groups. The main classifications of carbohydrates include monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides, these are the simplest carbohydrates, consisting of a single sugar molecule. Monosaccharides are classified based on the number of carbon atoms they contain. Trioses, tetroses, pentoses, hexoses, and heptoses. Disaccharides. Disaccharides are composed of two monosaccharide units linked together by a glycosidic band. Common disaccharides include sucrose, lactose, and maltose. And oligosaccharides. Oligosaccharides consist of a small number, typically 3 to 10, of monosaccharide units, linked by a glycosidic bands. They are found in certain foods and play roles in cell recognition and signaling. In polysaccharides, polysaccharides are complex carbohydrates composed of many monosaccharide units joined together. They serve as energy storage molecules, example, starch in plants, glycogen in animals, and structural components, example, cellulose in plant cell walls, chitin in fungal cell walls, and exoskeletons of arthropods.